Welcome back. First, some updates. So, by now, I've gotten from all of you the Pocket Mouse Lab, the Antibiotic Resistance Lab, and Unit 3 Homework 1. Most of you have turned in the reflective writing. If you haven't, then please do so immediately. And same goes for the Finch Lab. So, a few upcoming assignments. We're going to have Homework 2. I'm going to be posting that today. On next Tuesday, we're going to be doing the Human Evolution Lab. And then after that, you'll have the key terms list due, and that'll serve as homework 3. I'll be posting that today as well, so you guys can start filling those out and studying them immediately. So then next Thursday, we're going to have our Unit 3 quiz. That's going to be on April 9th. Something that you'll need to do today or in the near future is download this Respondus software. Okay, and there's a link on Moodle for this under today's class folder. So find that, download it, and you'll actually use this browser to take the future unit quizzes for this class. So I'm going to give you some additional reminders about this quiz and about downloading the software. And then again, for the quiz, our key terms list will be posted today, so go ahead and start completing that. And that'll help you to study for this quiz. Okay, so for today, what we're going to cover, first I'm going to review the lab assignments and the homework that we've done so far. Just go over those answers, tell you basically why the right answers are right and why some of the other answers are wrong. I'm going to dive a little further into phylogenetics then, and then we're going to talk a little about microbial evolution and some really interesting details in that regard. So first of all, let's go over the Pocket Mouse Lab answers. So question 1, what was the trait under selective pressure in these mice? So I'm sure you guys all remember that the trait under selective pressure in this case was the color of the fur of the mice. Okay, question 2. So this one was a little tricky, but it's something that you guys need to remember, and it's basically covers a common misconception for evolution. So the question is, did the change in terrain color cause the mutation in the mice? So we're talking about something that's causative of a mutation. And so the answer to this is, of course, no. Mutations and variations in traits do not arise due to selective pressure. So this is one of the most important concepts that we're going to cover in this whole unit. Mutation does not arise due to selective pressure. So remember, that's Lamarck's theory, and it's been basically proven wrong. So the mutation occurs randomly, and because it's a favorable trait, it became more common due to natural selection. So mutation is a random event, but natural selection is not. So this comes back again to the 4 forces of evolution, right? So natural selection is the only one that's actually an adaptive force. Mutations, random, gene drift, and gene flow are also random, but then they are acted upon by natural selection, which is non-random. So that's an actual adaptive force of evolution. Okay, so question 3, what gives the mice with dark fur an advantage over lighter colored mice? This was because the mice with dark fur were able to blend in with the volcanic rock, and thus their camouflage from predators. And then what is the selective pressure in this scenario? That's, of course, predation, right? So the mice that lack camouflage are more likely to be killed by predators. Okay, question 5, what gene was identified as mutated in the first population of mice researched? In the first population, the researchers identified a mutation in the MC1R gene, which appeared to be the cause of the dark fur color. And so, just for you guys' information, the MCR1 actually encodes a melanocortin-1 receptor, and this controls the activation of melanocyte cells that produce melanin. So this is relevant again to like the human skin color evolution that we talked about. It's actually one of the genes in humans as well that controls skin color and pigmentation. So that's really interesting. Okay, question 6, what did they discover when looking at mice from a different region of the Southwest with similar lava flows? So they found that these mice had adapted similarly. They also had these darker coats. However, this trait had arisen through mutation of a different gene. And so what this demonstrates, again, is that mutation, the process of mutation is random, but natural selection is not. So similar adaptations can be favored in these different regions, even though they originated from different genes. So that's really cool. And then question 7, since mutation drives evolution, the question is, do you think that most mutations are beneficial to an individual organism? And actually the answer to this is that most mutations are actually harmful, and that's because natural selection has already produced populations with favorable traits for their current environments. So what can make mutations beneficial in most cases is when environmental changes occur. So in the case of the mice, this is the lava flow that changes the desert surface color. And in that case, certain traits that may have otherwise been harmful, and so dark coats against the light sand normally would have been harmful, but these traits can quickly become beneficial after these environmental changes. Okay, so moving on to the antibiotic lab answers. So first, a superbug is a strain of bacteria that's resistant to multiple drugs that are commonly used to treat it. I don't think anyone got that one wrong. Number 2 and 3 deal with this diagram down here in the bottom right, which was on one of the pages. And so question 2, the diagram on page 2 shows how a bacterial population can acquire antibiotic resistance. And the different colors of the rectangles here, the question is, do they represent the different antibiotics used in the experiment? The answer to this is false, because these colors here do not represent the different antibiotics. They're representing the different species of bacteria that's present here, and basically bacteria with different traits. And so what you can see is, over the course of time, due to the exposure to antibiotics, some species of bacteria basically are dying off because they don't have the right traits to deal with these antibiotics. And so hopefully, you can figure that out from these types of diagrams. So the next question, in this diagram, the red bacteria survives antibiotic exposure, right, and yields new bacteria that are different shades of red. So what do these different shades now represent? So option A, bacteria from different families that have survived after the single red bacteria cleared the antibiotics. So this answer is wrong because a single bacteria is not going to just go around and chew up all the antibiotics in the environment. It's going to probably pump out the antibiotics that it's exposed to, or have some other mutation that allows it to survive it, but it's not going to clear the antibiotics. That's why all these other guys die. B, bacteria that are slowly dying from the antibiotics. That's wrong because these bacteria are able to grow actually because they're all now resistant to the antibiotics. So option C is the right answer, that natural variation in the population has now arisen from this single bacteria due to naturally occurring mutations. And so that's what the diagram is actually trying to show, is that over time, despite the fact that you had one survivor that produced more bacteria, these guys are going to be making subtle other mutations, right, that could enable basically future traits to be selected for. And then D, bacteria in various stages of the life of the cell cycle is just kind of random throw-out answer. Okay, so question 4, bacterial populations can occasionally acquire drug resistance very quickly because of which of these? And so horizontal gene transfer, this study talked about this, or the ability to pass genetic information between two different bacteria on plasmids. So that's correct. B, fast doubling times for bacterial populations. That's also correct. So bacteria replicate very quickly. And then C, only the bacteria that are resistant will survive the drug treatment. So that's also accurate. And so when you think about the Finch lab that you did, the researchers noted that it was kind of like very quick evolution that they observed after the drought, and that was because only the finches with the bigger beaks survived, right, the drought. And so that can be a cause of really rapid evolution, is when there's a lot of members of a species dying. So all the above is correct for this question. Number 5, I'm going to skip number 5. I think you guys all got that one right. And then number 6 is very important. So choose the answer that places the events leading to bacterial evolution in the correct order. So this is going to be the case for basically any question like this. So random DNA mutations occur first, always, right? Then you have antibiotic exposure, and this is the selective pressure that acts on these random DNA mutations in order to cause bacterial population evolution. So A is the correct answer here. So B is wrong, for example, because while random DNA mutations do occur first, the bacterial population is not going to evolve without the antibiotic exposure in order to place selective pressure for that DNA mutation. C is wrong because if you have antibiotic exposure first, then random DNA mutations, basically these DNA mutations are not going to be able to occur if you have the antibiotic exposure that acts on a completely susceptible population. They're just going to all die off in that case. So you have to have the random DNA mutations first. And again, this is because mutations do not arise due to selective pressures, right? Rather, they have to pre-exist, and then natural selection can act on them. Okay, number 7, how might mutations in these two bacterial proteins, GIR-A and PAR-C, cause resistance to quinolones? So this came from this table down here in the bottom left, GIR-A and PAR-C. And so what you see is that they're finding mutations in certain amino acid positions of either protein. So the answers are that quinolones bind the GIR-A protein near amino acids 83 and 87. That's likely because, if mutations in these positions are conferring resistance, then probably that's where the antibiotic binds near. And then again for PAR-C, near 80 and 84. So A and B are true, but C, these proteins destroy quinolones. There's really no evidence of that. And so A and B are likely, but not C. Then question 8, so some people missed this question, but it was pretty simple. What are antibiotics used most extensively for in the US? This question came directly from this pie chart that shows that the vast majority of antibiotic uses in the US is for non-therapeutic purposes in livestock. So this was right in the superbug study, pretty easy to find. So basically, yeah, just an easy question to answer. Okay, let's go through homework 1 real quick. So which of the following are adaptive forces of evolution? So again, this is right in your slides. Natural selection is the only one of these that's adaptive. The rest are somewhat random. So mutation occurs randomly. Gene drift is just random allelic changes that occur within a population. And then gene flow just occurs when you have two populations mixing together. So only natural selection is actually adaptive. And while all of these others can create variation for natural selection to act on, only natural selection in itself is an adaptive force of evolution. So natural selection is the correct answer here. Number 2, mutations arise based on needs imposed by selective pressures. You guys should all know by now that this is false. Mutations arise randomly. Mutation does not arise based on need. So that's an important one. Number 3, mutation by itself is a strong force of evolution. This is false because mutation by itself is random and mutations are oftentimes harmful. Pretty much just as often or more often than they are useful, mutations are harmful. So mutation by itself is not a strong force of evolution. When you couple mutation with other forces like natural selection, it can become a strong force of evolution, but by itself it is not. Evolution occurs only due to species adapting to their environment. So the answer to this is false, and that's because in some of these cases, like with mutation, gene drift, and gene flow, these can be occurring without natural selection, and the species aren't necessarily adapting to their environment. But remember, there can be random changes in the allele frequencies within a population, and that's what defines evolution. So evolution can occur somewhat randomly, so it's not only due to adaptation. Okay, number 5, human skin color gradually lightened as humans migrated further away from the equator. Why was lighter skin color selected for in these geographic regions? So answer A, these regions receive less UV exposure, so pigmentation is not needed to prevent skin cancer. So while this is true, if you watch the video, the researchers pointed out that skin cancer actually generally happens after like the age at which you're reproducing. So this probably wasn't a very strong force of this because people have already they've already basically done their reproduction by the time that they'd be developing and dying of skin cancer. So instead, they found that these regions received less UV light, so the lack of pigmentation, part 1, facilitates vitamin D production, which is important for effective reproduction. And then pigmentation also prevents the breakdown of folic acid, which in these regions with less UV exposure is not as important. So it's about this balance between both preventing the breakdown of folic acid and making enough vitamin D. So the answer to this is B and C, but not A. And you guys should remember this for the quiz. Okay, question 6. You treat a bacterial infection with chloramphenicol, an antibiotic that inhibits bacterial translation. This part is actually not very important. So in this case, all of the bacteria have ribosomes with the same amino acid sequence, which is sensitive to chloramphenicol. So this is the part that's important. All of the bacteria in this population are sensitive to chloramphenicol. So what's going to happen during your antibiotic treatment? The right answer is that all these bacteria are going to die. So in order to evolve resistance to chloramphenicol, the bacteria would have had to have a resistance gene within the population already. They didn't have that trait, so they're just all going to die. And so this is basically why we use antibiotics, right? In most cases, we don't have resistance traits in the bacterial populations that we're treating. So usually when you use antibiotics in a person, you have a good outcome in which all the bacteria die. Okay, question 7. Lamarck understood that mutation underlies variation and that this does not arise due to need. That's false. So Lamarck thought that mutation could arise due to need, right? So that's what we call the idea of Lamarckian evolution. When you have these mutations arising due to need, so when you think about Lamarck, you can think about like the horse turning into the giraffe because it needs to reach higher vegetation. So we know that that doesn't actually happen. So Lamarck didn't understand that, but he was important for eventually understanding that. Okay, number 8, the founder effect is an example of which force of evolution? So the founder effect is a type of genetic drift. Pretty simple. Okay, so that's it for homework questions. So let's move on to phylogenetics. So we touched on phylogenetics in Tuesday's lecture. And so what you see here is a phylogenetic tree representing evolution over time. And so these nodes basically within this represent species that are basically the last common ancestors between any of the species at the top of this chart. So I've labeled them with these 1, 2, 3, 4 here to make them stand out a bit more. So these normally won't be labeled right on evolutionary trees, but I've done this just for this exercise to make them more obvious. So the nodes represent species. And these species are the last common ancestors between, so like node 1, for example, is the last common ancestor between A and B. So in order to get kind of a deeper understanding of how these trees work, I'm going to go through a series of misconceptions, common misconceptions about phylogenetics, and then I'm going to correct them. And so at the end of this, hopefully you'll understand these trees pretty well. And so these are also questions that I am very likely to use on the quiz. So I might just give you, like, for example, a true or false question that has a misconception in it. And if that is the case, then you should answer that as false. So remembering these misconceptions and the correct way to interpret this is a pretty easy way to get some easy points on the quiz. Okay, so first misconception is that primitive species give rise to advanced species. So this is the idea that basically like the species down here or this species here at node 2 are more primitive than the what you'd call, in this case, more evolutionary advanced species. So this is wrong. This is a misconception. The truth is that species are constantly evolving to their specific environments. So none are really more primitive or advanced. So this species down here, or this node at 2, was probably quite highly adapted to its current environment. And the reason that it may have undergone this speciation process could be an environmental change or separation of two different populations. But that doesn't mean that this organism is any more primitive than either of these. So this is a misconception. Okay, our next misconception is that C and D are more closely evolutionarily related than C and E. So this kind of seems like it would make sense, right? I mean, C and D are closer on this chart than C and E are, but that's actually a misconception, and in truth, C and D actually just share the same last common ancestor as C and E. So as you can see, the last common ancestor between C and D is here at node 3, and the same is true for C and E. So while D and E share a more recent last common ancestor than C and D. The truth is that the relationship between C and D and C and E is basically the same. That's all you can interpret from the data presented here, that they share the same last common ancestor. Okay, next misconception is that species B here evolved from species A, species C evolved from species B, and so on. And so basically the way that you'd arrive at this misconception is by reading this from left to right. And so that is an incorrect way to read it. So the actual relationship here between A, B, and C is that A, B, and C all arose from the same last common ancestor. And so that's down here at node 2, right? So node 2 gives rise ultimately to A, B, and C over evolutionary time. And then A and B share a more recent last common ancestor here at node 1. And so that's the correct way to interpret basically the process of evolution within these trees. So don't read them from left to right, read them over time. Okay, the next misconception is that no evolutionary change between the very bottom of the tree, so down here at the very stem of this tree, and node 2. So this is one species, right? This line over time represents one species, and it doesn't undergo speciation until here at node 2. So the question is whether this organism is evolving during this time. And the answer is that yes, actually it is evolving. And so it's important to remember here that evolution and speciation are not the same. So species are constantly evolving, right? And evolutionary change occurs along each vertical line. So as time was going on, this species was definitely evolving, right? And so it's important to remember that the definition of evolution is just a change in allele frequency in a population. So of course this organism was probably evolving, either undergoing just kind of random genetic drift or other processes, but speciation occurred only up here, and that was likely because of a change due to a change in its environment, or maybe two populations became isolated from one another long enough to produce new species. So evolution and speciation are not the same. Okay, next misconception. Because there are more nodes separating A from E than there are A from C, there's more evolutionary distance between A and E than there is between A and C. Okay, so what do I mean by this? So you can see between A and E, how many nodes are between them? We've got 1, 2, 3, 4 nodes between A and E. And then between A and C, how many nodes are there? There are only 1, 2, 3. So fewer nodes between A and C. So does that mean they're more, basically more evolutionarily related than A and E are? And the answer is no. So the only proper measure of evolutionary distance between species is the time since the last common ancestor. So the last common ancestor between A and C is here at node 2, and that's actually the same as the last common ancestor between A and E. So when you consider that, the distance between A and C is the distance between A and node 2. So it's this vertical distance basically. And that's the same vertical distance as between E and node 2. And so basically these have the same amount of evolutionary separation, despite looking like they're in closer proximity to one another on the tree. So hopefully that makes sense. So the only measure of evolutionary distance on these trees is the time since the last common ancestor, not their physical distance from left to right. Okay, so that's basically the misconceptions that I wanted to cover for phylogenetics. The last thing that I want to mention is that it's important to remember that sorry about the blurriness, but for all kingdoms of life, they're thought to have all arisen from a single-celled species of organism billions of years ago. So one species is thought to have given rise to basically all species on Earth at this point. So the idea is that we can trace the evolutionary history of any two species on Earth and find a last common ancestor between them. So that's another thing that's important to remember about these phylogenetic trees, is that everything can be traced back to a single last common ancestor. That's the theory anyway. Okay, so while we're thinking about that kind of microbial evolution into multiple species, let's talk about microbial evolution. So I'm sure you guys know that there are a lot of different types of microbes out there, right? There are bacteria, there are viruses, there are fungi, and there are multiple species of all of these. And really, there is different from one another as we are from any other like animal that you can think of on Earth. So what are some of the selective pressures on microbes that lead to their evolution? Well, there's of course their environmental conditions, just like us, right? So microbes live in very diverse environments. You have microbes in the ocean, microbes that live on your skin, microbes that live in your gut, microbes that live in the soil, kind of just almost everywhere you can think of, there are microbes on Earth. Another selective pressure, of course, is nutrient availability. So microbes, just like us, have to eat, have to produce energy. And so the availability of nutrients to them can be a massive selective pressure, just like us, just like the Galapagos finches, etc. Then, of course, competing microbial populations. So microbial populations often exist among one another, and so they will have competition for resources and space. And then additionally, some microbes will actually create antimicrobial compounds to kill off other microbes in their surroundings. So you may or may not know this, but the antibiotic penicillin, the first one ever discovered, was actually discovered from a microbial fungi species. So clearly, there's a lot of competition between microbial populations. And then if microbes have to live on or in the host, then the host can basically create pressures for them, such as the host immune defenses, or in our case, in the case of humans, we often treat bacterial populations with antibiotics or other drugs. So this is another example of pressure that we put on our microbial populations. So how do microbes basically adapt and evolve to these? So first of all, of course, there's random mutations. This happens in all microbes, bacteria, viruses, fungi, etc. There's horizontal gene transfer that occurs mostly in bacteria. And so bacteria are capable of receiving genetic information from other microbes in their environment. And so this can come from circular DNA called plasmids. So this is what we talked about in the superbug lab. Then there can be recombination that occurs with closely related pathogens. This mostly happens in viruses. And so this is something that happens sometimes in flu, and basically it can create new combinations of genes and allele within the population. And so this is something like in flu, for example, that can allow it to jump from birds or like pigs more easily into humans. These recombination events that create new combinations of genes and alleles within the virus. Then there's the fact that microbes basically grow very rapidly. They have fast doubling times. So bacterial populations can expand in basically just a few hours' time, and this is even faster for viruses. So microbes actually evolve much faster than their hosts, and this can put a lot of evolutionary pressure on hosts. So this creates sort of this evolutionary arm race between hosts versus pathogens. And so there's this Red Queen hypothesis that exists that basically states that in reference to an evolutionary system, continuing adaptation is needed in order for a species to maintain its relative fitness amongst the systems it is co-evolving with. And so why is it called the Red Queen hypothesis? This actually comes from Alice in Wonderland, interestingly. And so the excerpt is basically Alice is in this race with the Red Queen. And the Red Queen tells her, now here you see it takes all the running you can do to keep in the same place. If you want to get somewhere else, you must run at least twice as fast as that. And so this is kind of referring to the constant evolution that both the microbe or the pathogen and the host has to be undergoing in order to maintain their fitness relative to one another. So in this way, pathogens can shape the evolution of their hosts. And so more evidence of this is that immunity-related genes are among the fastest evolving in the human genome. So one of the types of genes in humans that evolves the very fastest are these immune genes, actually. And so we're constantly trying to keep up with the microbes that are attacking us, basically, is what that demonstrates. And then another way that pathogens shape our evolution is that the human genome, it turns out, is actually around 8% of it is viral derived, which is a really massive amount of genetic information when you think about it. So how does that happen? So you guys may be familiar with the idea of certain viruses that can actually jump into our chromosomes, insert their genetic information into our chromosomes. So that's something that like HIV does, for example. It's a retrovirus. So when when viruses do that and actually stick around over evolutionary time and kind of become just part of our DNA naturally, we call those endogenous retroviruses. So most of these are actually epigenetically silenced, and so they don't generate infectious particles. So they're not like producing infectious virus in our bodies. But some are actually co-opted to serve purposes for us, which is super interesting. So one example of this is there's a gene that's expressed in neurons specifically. It's called ARC, and it's been discovered that this gene is actually a repurposed viral capsid that now is used to package mRNA, and this is actually super important for brain development. And so the gene product of ARC is actually these proteins that self-assemble into viral capsids, and this is actually like a cryo-EM picture of what this capsid looks like. So these capsids are actually produced by your neurons all the time, and they package mRNA and then are secreted by your neurons and can like transmit information between your brain cells, which is really crazy. And this all comes from an ancient virus that's no longer a functional virus, but now serves our purposes. And then many retroviral genes are also have been found to be important for pregnancy, and there's probably a lot more that remains to be discovered in that regard. So that's just super cool stuff. You don't necessarily need to know these things for the quiz, but just cool stuff that I thought you might like to know. So in terms of thinking about this co-option of microbial components, another way that this happens is actually symbiosis. So you guys have probably heard about symbiosis, or the idea that basically two organisms can coexist and both benefit from the relationship. So for example, within the human body, so all of us have about 10 times more bacteria on and in our bodies than the actual number of our own cells, which is crazy to think about. And within our gut microbiota, so our gut is colonized by a lot of types of bacteria, and these are crucial for digestion. They also help protect us from infection by like pathogenic bacteria, and they also may be important for brain development and a number of other things that occur within your body. And so the bacteria also benefit from this relationship because, of course, they just kind of get to hang out in a nice niche, and we feed them all the time. And so it's this really great symbiotic relationship where both of us benefit. Another example of this symbiosis is actually mitochondria. So you may or may not have ever heard of this, but we all know what mitochondria are, right? They exist in all of our cells, and they're crucial for the function of eukaryotic cells. So we've all heard before that mitochondria are the powerhouse of the cell. People say this because mitochondria are one of the major producers of ATP within our cells, and this is a super important molecule for cellular energy, basically. And as we know, mitochondrial mutations can be bad news. They can be fatal in some cases. So it's clear that mitochondria are, crucial for the functions of our cells. So where did mitochondria come from is the question. So, the mitochondrial origin is actually predicted to be from bacteria, and so the reason for this is that mitochondria actually contain their own genome. So we've talked a little about this, that mitochondria have their own set of DNA, and mitochondrial DNA, it turns out, actually looks a lot like bacterial DNA. So they have a circular DNA genome, and they have a lot of genes that are kind of homologous to those in bacteria. So in the 1970s, when some of this started being discovered about mitochondrial DNA, researchers came up with a mitochondrial origin theory in which they think that ancient specialized bacteria were actually endocytosed or taken up by an ancient eukaryotic cell. And then because this bacteria provided energy from the cells, the cells likely in turn provide protection for the bacteria. And so this creates a symbiotic relationship. And so over evolutionary time, mitochondria have actually been completely co-opted, and they now just exist in basically all eukaryotic cells and help to power them. And so this is a really crazy and important idea of this symbiosis between a eukaryotic and a prokaryotic cell, in which a bacteria was actually taken up and then co-opted for cells to serve purposes basically for both of them. So some key takeaways about microbes and human evolution. So microbes and their hosts both exert selective pressure on one another. Pathogens and hosts are constantly co-evolving, so that's that Red Queen hypothesis. And then many microbes also exist in symbiotic relationships with their hosts. So some examples of this are gut microbes and mitochondria, and keeping in mind where the mitochondria actually came from. All right, so that's it for today's lecture materials. So there's one action item for today, something that you should do immediately following this lecture. So go onto Moodle and go ahead and download this Respondus browser. So there's a link to a website where you can download that on Moodle. It'll take you right there, and you just hit the download button, and that'll be underway. So the reason you need this is because we're going to take future unit quizzes using this browser. Basically, it just kind of like locks your computer into one function, so it's not going to allow you to like look at your notes at the same time, for example. So over this weekend and next week, take a look at the Unit 3 Homework 2. You can take your time. Remember, just take your time while you're answering these. Look for the answers in the slides. They're not going to be like trick questions. They're just going to be questions that you need to understand basically for the quiz. Then I'm posting also today the key terms list. So you guys should work on that, get started on that, work on it over the weekend. That'll be due next week before the quiz. That'll serve as your homework 3, and it'll also, of course, be a nice study guide for you. Then next week, Tuesday, we're going to talk about human evolution. Basically the fossil record and order of events that occurred within human evolution to kind of differentiate us from our last common ancestor with chimps. And then we're also going to do a human evolution lab activity. And then Thursday I will post a review video that's basically just going over the key terms list and things you should know from that. And then I will have, before the class time, an optional question and answer session via Zoom, so you guys can pop into that at any time if you want. I'll probably start that at like 9 a.m. And then we're going to have the Unit 3 quiz. And we're going to have the quiz, it's going to be open during the normal class hours. So I sent you guys an email about this, but just, another reminder. So the quiz is going to become open at 10:30 and it'll close at 1 p.m. So you need to make sure that you're able to take the quiz within that time. If you're not, then you need to let me know ASAP. And that's all I've got. So see you next time.